Hello, my name is Dr. Jennifer Koenig, and we're going to be talking today about interethnic differences and pharmacokinetics. So the aims of the project were to carry out a scoping review of the literature and to review how differences in diet and human physiology and biochemistry between ethnic groups were different and how this can affect pharmacokinetics. We were then going to use that information to develop guidance for academics teaching pharmacokinetics to help them address this diversity and think about how to teach it, to use the findings to produce research-based learning materials, and then finally to evaluate their use. So we know that there's a lot of inter-individual variation in pharmacokinetics. We know that humans respond to and handle drugs very differently, and there are quite a lot of reasons behind this. But there is actually quite a literature around the role of ethnicity in pharmacokinetics, and we were sort of intrigued by this and wanted to find out more about it and think about whether or what we should be doing in our teaching. Before we go on, we really need to think about some of the definitions of these terms, race and ethnicity. So race has typically been used as a sort of a taxonomic categorization, so largely sort of skin color, hair texture, sort of superficial appearance, really, to divide people into groups. It's important to note that the term race is a social construct and there's no biological genetic basis behind it. We need to be really clear about that, I think, with our students. To be fair, most of the pharmacokinetics literature really talks about ethnicity. And ethnicity has a lot of different definitions and can be used in different ways, but typically there are elements of identity that are inherited through ancestry and culture rather than just appearance. Ethnic groups can be very broad, so for example whites or Asians or so on, but some of the papers looked at smaller groups, so they might have had Japanese and Korean and East Asian and West and different, subdividing up the Asian groups into smaller ones. So the other thing that's important to note is that there are no internationally accepted criteria. So different countries around the world will use a different set of ethnic group tick boxes to choose from, and some countries don't actually collect ethnicity information, so it may not actually be available. So I'm going to hand over to Shola now, and she's going to take you through some of the findings from the scoping review.
So in the next couple of slides, I'll take us through some of the key findings from the research that we've conducted on the subject of interethnic differences in PK, and we're going to be looking at what we found in literature with respect to this particular subject and what colleagues in higher education or educators in higher education currently teach with respect to interethnic differences in pharmacokinetics. Next, we looked at the papers that observed interethnic differences in pharmacokinetics, and what we were looking for in these papers was, first of all, to identify the ADME processes that were associated with the PK differences that were reported in those papers, and then we compared what we observed to the responses from our respondents. So we compared it to what our respondents believed were the PK processes that was associated with interethnic differences in PK. And our comparison reviewed that metabolism accounted for the main PK process that was attributed to interethnic differences in pharmacokinetics. Our respondents at the time of the survey were educators in industry or in the academia, and they had either taught PK in the past or were currently teaching PK. And the scoping, the survey was basically just to gain some insight on their attitudes towards teaching interethnic differences in pharmacokinetics. Then next, we looked at those papers that we identified as the papers that observed differences in PK between ethnicities, and we looked at the suggestions that the authors of those papers thought might have been responsible for interethnic differences, or they suggested might have been responsible for interethnic differences. And there were a range of mechanisms that were proposed, but genetic polymorphisms was the main mechanism that was associated with the interethnic differences in PK. Of all the papers that were scanned through, about 74 papers, 37% of them mentioned genetic polymorphisms as a potential reason for the interethnic differences in PK, but there were other reasons that were mentioned, like diet, physiological differences like weight, differences between ethnicities and blood flows, also biochemical differences like expression levels of certain enzymes and proteins differing between ethnic groups. Now let's consider one or two papers that identified non-genetic factors as potential reasons for interethnic differences in PK. One of such papers was this one, a global population PK analysis that was done for Olusola. And the authors of this papers concluded that there was a significant regional difference in PK of Olusola. They reached that conclusion based on the bioavailability of Olusola, which was found to be 52% higher in patients who lived in Asian countries compared to patients who lived in Western countries. So that suggested that the PK of Olusola was more to do with where the individuals lived rather than whether they were, Asians or they were Westerners. That observation was further, reiterated when they grouped patients based on their clearance, their oral clearance, and they found that Asians who lived in the West, they grouped similarly, with non-Asians who lived in the West. So it was a case of where they lived rather than whether or not they were Asians. So there are other factors that might have, environmental factors that might have affected the PK in that case, certainly not genetic factors. There might be factors like diet, for example, or other environmental factors that might have influenced the PK of this drug. Another example of when PK could be affected by non-genetic factors, for example, diet in this case, is with paracetamol or acetaminophen. We know that paracetamol, from this research that was done a while ago, the absorption of paracetamol is impaired, is slowed down in patients who take vegetarian diets compared to patients who do not take vegetarian diet. So in that case, if you have people who live in ethnic groups where vegetarian diet is, common or, is likely, is highly likely, then we might assume, therefore, that if those patients are actually, in fact, on vegetarian diet, the absorption of paracetamol may be impaired or may be slowed down in such patients. So a number of the studies we were found made mention or reference to diet as a possible reason for differences in PK between ethnicities. So another non-genetic factor that could in fact affect PK differences between ethnicities that I would like to highlight in our talk today is weight differences between ethnic groups. And this has been demonstrated in literature in a number of studies, but we are just going to highlight efavirenz and selumetinib. And what we see for efavirenz and selumetinib is based on these separate studies is that they were able to link the differences in PK between the ethnicities that were studied to the differences in the weight between the ethnicities that were studied. And we know, based on data that is out there, that the average weight of people who are Africans, for example, or Americans, or people who live in Southeast Asia or Western Pacific region differ from each other, and they definitely differ from the average adult weight that is recognized more generally, that is between 70 and 80 kg, a weight that is based on a healthy adult male Caucasian.
So what does this now mean then for teaching of pharmacokinetics, and how are we going to go about developing these teaching materials, teaching resources? So we wanted to think about some guidelines first of all, and one of the things we wanted to do was to discuss some of the poor evidence for linking these single nucleotide polymorphisms to ethnic differences, and really encourage students to break down and tease out some of the arguments that are going on here. In particular, the idea that the prevalence of the different isoforms is not absolute. So it's not that one ethnic group is 100% of one isoform of an enzyme and another ethnic group is 100% of the other. It's usually much more subtle than that. And that has important implications for practice. The other aspect is to be aware of the experimental design. So we've done a scoping review so far, and we've looked in depth at a small number of the papers, and it's been very interesting to do a critical analysis of experimental design, statistics, sample size, statistical power, and so on. And we really feel that these papers would be a very useful tool in helping to develop critical analysis in students. The other thing we wanted to be really clear about with this is to tackle some misconceptions. So let's be absolutely clear that the experts, geneticists' views, and this has been the case for many years now, is that multiple genes and multiple environmental factors interact in the development of most traits. And that if we look at the genetic diversity of the different ethnic groups, we can see that there's a much greater ethnic diversity amongst just Africans than there is amongst Europeans, and there's that the genetic diversity is overlapping between the different populations. So that's what the experts are seeing, but what we see in the high school biology textbooks and in the curricula as well is that most of the genetics that's taught is grossly oversimplified and focuses primarily on monogenic traits like sickle cell and cystic fibrosis, things that are often linked to particular racial groups as well in the textbooks. So what's happened really, and it's partly I think due to the curricula and the textbooks, and partly just due to the sort of prevailing discussion and media input, is that a lot of people have this misconception that ethnic groups show distinct genetic differences, when in fact they don't. So this has been termed genetic determinism. So many people have this misconception that it's our genes that determine us, it's the genes that are our essence, if you like, and really completely underestimate the effect of the environment on the traits and diseases. So it's really important that we have that in mind when we're teaching in this area. So our recommendations are that we should be discussing individual differences arising from a range of factors and include weight and diet, and as well as isoforms of transporters and enzymes. And that we should not use race or ethnicity as a proxy for a variable that could be measured directly, whether that's the proportion of people with a particular isoform of an enzyme, or whether it's weight or diet. We should be measuring those things and not just trying to infer them from ethnicity. So the next stage in our work is to develop the teaching resources now. So we're currently working on developing questions, exploring these, the basic pharmacokinetic concepts, but using contexts that involve inter-individual differences, so getting students to apply their understanding of pharmacokinetics in these contexts. And then the second area is to use some of these papers to develop critical analysis workshop so that we can encourage critical thinking amongst the students. So thank you very much for listening, and thank you to the BPS for their support.